Hello everyone, and thank you for coming to our webinar today. I'm Moya, the technical support of BMKGENE. As the technical support, I have been exposed to many RNA sequencing projects since I joined the company. Also, I would like to share my experience on how to start your first mRNA sequencing project. What I'm going to talk about today is divided into four parts as follows. The first part is about why we do RNA sequencing and what problems we could help you solve. The second part is about how we perform mRNA sequencing. And the third part is what biological information you could get from sequencing data. By the end, I would like to introduce the BMKGene service scope. And why do we perform mRNA sequencing? RNA is one of the most important molecules in molecular biology today and is the molecule that could be linked to the biological function information. RNA is mainly classified as mRNA, link RNA, saccharine RNA, and others. Currently, we are using NGS technology to sequence and analyze these RNAs or some of them. It mainly reflects the expression levels of them, quantifies RNA, and predicts the function of gene expression. However, our focus today is on mRNA, and with the development of NGS technology, RNA sequence has become more widely used. Firstly, in the field of mRNA biology, RNA sequencing could be applied to single-cell gene expression, protein expression, RNA structure analysis, and so on. Secondly, the concept of spatial transcriptome is also emerging. Of course, we could also provide service related to spatial transcriptome if you are more interested. In addition to this, for example, mRNA sequencing analysis could give us information about differentially expressed genes which could be used in different academic fields. For example, in medical research, mRNA-seq could quickly provide you with information about the causes of altered gene expression patterns in cancer genes or other diseases by comparing normal samples with significantly altered expression patterns in disease samples. Also, their functional analysis. And in agricultural research, RNA sequencing results could identify genes that affect specific plant type and could help you get a more comprehensive understanding of diversity and the complexity of the transcripts. And if you have a need for RNA sequencing in one of the academic areas mentioned above, how should we proceed? The following process is the solution proposed by BMKGENE. And this is our basic process of mRNA sequencing, from total RNA to library preparation and the sequencing and analysis. You would find that quality control is done between each step. This is because in a high throughput sequencing projects, in order to ensure the reliability of the final biological results, we must ensure that there are quality standards at each step to tell the reliability of each node. The following is the sample delivery standard for mRNA sequencing. Because it is directly related to the data output. If you want to have high quality data output, the most important condition is that the sample material is good, so as to get good results. For example, if the total amount is not up to the standard, it would be affect the library preparation experiment. Also, if the amount is too low, the concentration of the library would be too low to meet the standard of sequencing. And there is also a low ring level indicating our degradation, which results is uneven data distribution and a quantitative impact. In other words, only when the mRNA is mostly intact could we get a bad effect. The use of complete RNA is the key to success of downstream gene quantification, sequencing, and other experiments. Therefore, sending samples within the standard is crucial. And when we do QC internally, we generally use non-drop to metal concentration and binocular equivalent 5,400 test integrity. I shared two very representative good quality sample and the degrade sample on the right. The good quality sample is shown on the top, and its 18th peak and 28th peak are both very sharp. In the figure below, we could clearly see that RNA is seriously degraded. So we usually do not recommend to proceed such samples. After confirming the high quality samples started, the lab preparation experiment could also be proceeded. And mRNA sequencing could divide it into non-directional mRNA and directional mRNA. Yeah, and according to different library preparation methods. For the non-directional mRNA, because the connectors we added to both ends of double-stranded cDNA was symmetric. It was impossible to judge whether the detected range were from the positive or negative strand after sequencing of the obtained library. Since the expression of genes in RNA sequencing is calculated from the matter range, the expression of transcripts obtained by ordinary RNA sequencing is actually the sum of positive and antisense transcripts, and such results may not reflect the actual situation of transcripts. In order to avoid the problem that the ordinary RNA sequencing could not distinguish the direction of transcript, directional library methods emerged later. It could return the orientation information of transcripts during the sequencing. And for the majority of eukaryotes, we believe that the sample size required by non-directional library preparation is lower and sufficient to obtain complete genetic information. However, if you would like to a higher demand for quantitative information of samples or wants to clearly analyze the transcript information of predictive strand, we suggest the directional mRNA. Okay. And after the library preparation, sequencing could be arranged. We mainly choose NORA 6,000 platform available company for sequencing, and the sequencing strategy is P150. For most species, we generally recommend 6 gigabytes of data, which is enough to obtain relatively accurate and complete gene quantitative information. At the same time, a lot of genes could be recovered, but if you would like the clear sequence steps for your study samples or want to explore more abundant genes, we also suggest you to increase the amount of data. In addition, if your sample has a small genome, like 2 or 3 gigabytes of data is fine, and the data could be guaranteed in about 3 weeks. Of course, the actual turnaround time would depend on the number of samples or the sample type of the project. And so, what biological information can we get from the data that we have sequenced? Analysis of these parts of the content would be very complete to give the data certain biological significance. Here, I would like to use our standard analysis pipeline as an example to show you how we analyze the sequence data. Our analysis process is also divided into analysis with reference genome and without reference genome. And the biggest difference between them is the presence or absence of reference genome. I also roughly divided it into 4 parts, moving from raw data to clean data. The second part is moving from raw data to clean data that could be used for analysis is determining the quality and accuracy of sequence data. And the second part is when we don't have the reference genome, we usually assemble the data first. If the reference genome was comparison viable and the rates were directly selected for comparison with the reference genome. And the third part is sequencing structure such as alternative splicing, novel gene propagation, and so on. And the last part is expression quantification, which is also the most important part. including gene expression analysis, differential expression analysis, and the differential gene annotation. Yeah, and this part. And I'm going to give you an example of the analysis with reference genome, and I will show you some more important results of the analysis. And firstly, in the process of mapping, such information would be obtained to prepare for the subsequent quantitative analysis. Comparison efficiency statistics can directly show the matching degree between the sequence samples and the selected reference genome, and also directly reflects the use ratio of data. As shown in the right figure, since we measure the transcript mRNA, it belongs to the action region on the genome. So, in our comparison results, action region must account for the majority, and there are also two other regions. One is the intergenic region, and the other is the intronic region. And the reason is that some genes in the genome are not well defined, but it could also be new genes that have not been predicted, or some non-coding genes else. And all the followed analysis we did was based on the unique mapped rate. It is the number of rays aligned. It is and it is the number of rays aligned to the reference genome, and the quantification of RNA was carried out based on that. And in RNA sequencing, when we quantified gene expression, the index we used was RPKM, which was calculated in the following way. What was shown on the left is abundance of all the genes expressed from all the samples, and the content on the table is a very intuitive representation of expression of genes in different samples. After we have all the information from the gene table, the most important part of our job is to look for differences between genes. For differential expression analysis, the basic method is to calculate gene expression in its biological meaningful method, and then to find genes with statistically significant differences through statistical analysis of expression, so as to select the suitable genes and measure the reliability of the results. Usually, the differences in gene expression levels between two samples could be viewed through volcano map, because the screening conditions could be defined manually. We usually generally take for change greater than or equal to 2, and if they're less than 0.01 as a screening standard in the process of differentially expressed genes detection. And the correlation heat map of samples within and between groups were calculated according to the expression values of all genes in each sample. And the heat map was drawn to display the differences between sample and the reputation of samples within groups. The higher correlation coefficient between samples, the closer the expression pattern is. The core aim of enrichment analysis of DGs is to assign biological functions to the DGs find, which can be given by GU-OKGG. In conclusion, after finding the important gene by this analysis, the actual function of these genes could be learned. Different databases, differential annotation information, and different association analysis could be all reflected by the reflected the biological significance. And finally, I will show you the service scope of BMKGENE. BMKGENE has been actively engaged in innovation and developing high throughput sequence technologies and bioinformatics for over 12 years. sorry, we have built in most cutting-edge, high-throughput sequencing platforms, including Illumina, Nanopore, PepBio, 10x Genomics, and high-performance computing platforms. We have more than 500 professional corporation organizations and participated in more than 3,000 collaborative papers. Is a biotechnology company with 30 years of professional experience. And BMKGENE is committed to providing the most comprehensive services covering genomics, transcriptomes, and single cell omics, microbial genomics, and so on. And if you are more interested in services or have a need for the research results project, please contact our sales team or TS team for consultation. And that is the end of today's webinar. Thank you again and for listening and look forward to working with you. Goodbye.